Myself, Dr. BIBHUTI Dutta Singh, PG Department of Zoology, D.A.V.P.G College, SIWAN, BIHAR. Today, we are going to discuss about Golgi complex. This topic is most important for B.Sc. Part 1st Zoology honors. So, in this potential topic includes point number 1, introduction of Golgi complex. Point number 2, morphology of Golgi complex. Point number 3, ultra-structure of Golgi complex. Point number 4, chemical composition of Golgi complex. Point number 5, function of Golgi complex. Point number 6, origins of Golgi complex. Now, first of all, I discuss about the introduction of Golgi complex. Actually, the Golgi complex is a canalicular system which is composed of parallelly arranged flattened membranous sac, vesicles, and tubules performs certain important cellular function such as biosynthesis of polysaccharides, cellular etc. Golgi complex of plant cells and lower invertebrates is known as dictyosomes. Then after I discuss about the historical background of Golgi complex. First of all, Camillo Golgi 1898 first discovered of Golgi complex in the nerve cells of barn owl. Morphology of Golgi complex occurrence mostly all eukaryotic cells except certain fungi, sperms, mature sieve tubes and RBCs etc. Position: the Golgi complex is polarized in exocrine cells. It lies between nucleus and secretory pole. Shape and size. The shape and size of Golgi complex is related with secretory or metabolic activities of the cells. Number. Actually, the Golgi complex is variable from cell to cell and depend upon the secretory function of the cells. Then after I discuss about the ultra-structure of Golgi complex. According to Dalton and Friggs, 1954, recognized three membranous component of Golgi complex under electron microscope. Number 1, cisternae. Number B, vesicles. And number C, vacuoles. First of all, I discuss about cisternae. The cisternae is the flattened, elongated, parallelly arranged, smooth, membranous with shorn ends. The space between two cisternae is called cisternal space. The cisternae are separated from each others by a space is about 20 to 30 μm is called intercisternal space. The internal cisternal space contain a layer or parallel rods known as the intercisternal elements. Then after I discuss about vesicles. The small drop-like structure and droplet-like structure is about 400 angstrom to 800 angstrom in diameters. These are pinched off from the tubules of cisternae. These are two types. Point number 1, smooth vesicles and coated vesicles. These are smooth vesicles, these are a smooth surface, while coated vesicles and these are rough surface. Then after I discuss about the vacuoles, the large rounded sac-like structure and filled with the amorphous or granular substance. These are formed either by the expanded cisternae. Thereafter, I can discuss about the chemical composition of Golgi complex or biochemical composition of Golgi complex. According to biochemical analysis, and they are made up of phospholipids, that is lecithin, amphiphylline, and protein membrane, etc. Protein enzymes and Golgi complex shows and great variations in protein and enzymes. Some are, that is ATPase, ADPase, CTPase, or TPPase. Carbohydrates, glucose and monos, galactose, etc. Vitamins, Golgi complex stores vitamins and liberates slowly in the cytoplasm. Then after, I discuss about the function of Golgi complex. The important function of Golgi complex can be as followed. 1. Absorption, storage, and transportation. Golgi complex is found to be more developed in secretory cells, and this indicates the main function of Golgi complex is absorption, storage, and transportation of secretory products. Then after, the function of Golgi complex, formation of cell wall and cell plate. Actually, the cell wall of plant cell is formed by the Golgi complex, the cell plate which formed during the cell divisions. Next, the formation of acrosomes. The Golgi complex is responsible for the formation of acrosomes during spermatogenesis. Actually, the spermatogenesis is the process. By this process, the formation of a sperm, and this process is known as the spermatogenesis. Another function, formation of lysosomes. The primary lysosomes are formed by Golgi complex and Golgi cisternae in the same ways as the secretory vessels. Another function, and pigment formations. The Golgi complex has been described as the site of pigment granules, that is melanin, and which is responsible for the coloration of eyes, hair, and skin is black. Next function, formation of plasma lemma. Membranes of the vesicles produced by Golgi apparatus and join in the regions of cytokinesis to produce new plasma lemma. Next function, synthesis and secretion of polysaccharides. The mucin secreted by goblet cells of clone contain proteins, carbohydrates, and sulfates. This is synthesized and secreted by the Golgi complex located between the nucleus and free surface of goblet cells. Another functions, the lysosome formation. The Golgi complex, the important role in the formation of lysosomes. The process of lysosomes are lysosomal hydrolysis, that is hydrolytic enzymes synthesized by the ribosomes located on endoplasmic reticulums to endoplasmic reticulums. Endoplasmic reticulums forms small vesicles around them, and those vesicles are transformed into the Golgi complex. A storage of proteins. Vacuoles and vessels, which are the main components of Golgi complex, becomes filled with the protein lipid materials for the storage, and this stored produced help in the secretory action, helps in enzyme formation, and Golgi complex as a great intracellular center of enzyme formation. Glycoprotein and glycolipids. Protein synthesized by the rough endoplasmic reticulums and lipids synthesized by the smooth endoplasmic reticulums and reach the cisternae of the Golgi apparatus. Here, they combine with the carbohydrates to form glycoproteins and glycolipids. Then after the last one, I discuss about the origins of Golgi complex. Actually, from endoplasmic reticulums, some biologists and cytologists regarded the Golgi complex as a differentiated part of the endoplasmic reticulums. The structural continuity between endoplasmic reticulums and Golgi membranes have also been observed. Next, the formation and the Golgi complex and from plasma membrane. Golgi complex nearest to the endoplasmic resemble. It is a structure and chemistry, while and those resemble the plasma membranes, indicating a polarity of organelles. In the biological export of secretions, small vessels from Golgi complex with the plasma membranes extruding secretions into the surface of the Golgi cells. Membrane then becomes part of the plasma membrane. In one scientist, Daniels, described the origin of Golgi cisternae from the plasma membrane in Polymexa. From nuclear envelope in brown algae, Golgi complex originated from nuclear envelope. He noticed the origins of vesicles from the nuclear membrane, which resembles the vesicles present on the formative phase of Golgi complex. Last one, the conclusions. After going through the above discussions, the Golgi complex is a tubular structure and mainly responsive for function and responsive for secretion and transportation of molecules and synthesis of polysaccharides. Thanks.